I'm Aran Nayebi, and I'm a new assistant professor here at Carnegie Mellon. Today, I'm going to give you an overview of my work at the intersection of machine learning and neuroscience. One of the great sources of motivation for me is that humans and animals exhibit a range of interesting behaviors in dynamic environments. So, for example, you and I can rapidly recognize the objects in the scene under a variety of viewing conditions. And furthermore, we can also parse the relations between objects, namely that buildings and trees line the sidewalk, cars are on the road, all of which constitute a rich understanding of the scene. Furthermore, animals can exhibit complex multi-step plans. So here you see a crow trying to grab a reward that it couldn't otherwise reach with its beak. Furthermore, animals can navigate novel environments. So what you're seeing here is a top-down view of a rodent navigating an open field, and the red dots are place cells that fire in the hippocampus of the animal as it enters a particular location in space. Finally, animals can flexibly and masterfully interact with their environment, greatly rivaling any robot that we have today. So for me, the overarching question that guides my research is, what are the core design principles that give rise to these abilities? And we know that this is hard because we don't have any handwritten rules that tell us how the brain reformats dense sensory information to give rise to complex behavior. So how do we bridge the gap from neurons to behavior and scale to the messy and challenging environments that humans and animals naturally face? Only in the past decade have we gotten a handle on this question in the domain of scene understanding, especially in vision. In particular, it's known that the primate ventral stream implements object recognition, whereby an image, like this picture of Marie Curie, enters the retina and then proceeds down a hierarchy of areas that process it from V1 to V4, all the way up to IT, or inferior temporal cortex, whereby object recognition judgments that this is a person rather than a cat or a dog are readily read out. And convolutional neural networks have been shown to be a very good model of the system, specifying a mechanistic wiring diagram that goes from inputs to object category outputs. And in particular, they're actually inspired by visual neuroscience, specifically the hierarchy and the retinotopy that it exhibits. And furthermore, we can view them as functional models, one, of the ventral stream, in that they perform this object categorization behavior, at least in the first 250 milliseconds. And furthermore, and what's most interesting to me is that they're also structural models of the system, in that if you optimize a CNN for a task and then fix the filter values and then evaluate them on new held out images using linear regression on the activation patterns, we can actually predict these individual neural responses in the real biological system well. So if we take this linear regression neural predictivity to IT on the y-axis, and on the x-axis report the ImageNet top one validation accuracy of each model, where each dot here is an individual CNN, what we see is that there's a strong relationship between these two measures. In particular, what we see is an alignment between a neuroscience goal, namely predicting variability in neural response patterns, and an AI goal, performing this object categorization task well. So we can ask, is this an accident? Did we get lucky? Or is this emblematic of a deeper operating principle? Well, going back to this picture that I was just showing you, I would just want to highlight quickly the kind of the four ingredients that give rise to the CNN model. One is the architecture class, the convolutional neural network architecture. Next is the task that we're optimizing on, 1,000-way categorization on a data set like ImageNet, for example, consisting of millions of naturalistic images. And then finally, optimizing this task through this architecture on this data set using a learning rule like backpropagation. So why am I highlighting these four ingredients is because each of these four ingredients has a direct readout to the four conceptual axes by which we can get a systems level understanding of a brain area. Specifically, if I list all of these four basic machine learning ingredients on the left, each of them has a direct readout to some corresponding component in neurobiology. In particular, the architecture class is a hypothesis for the circuit neuroanatomy of the system under study. The task is our hypothesis for the ecological niche or behavior that is thought that brain area to perform. The data set constitutes the sensory diet or the environment in which that brain area has been evolved under. And then finally, the learning rule plays a role of efficiently performing a sort of natural selection and developmental plasticity to get us to the adult state of the animal. So if I put those same four knobs just on each of the four corners of this slide, the confluence of these ingredients gives rise to a task-optimized artificial network, which we then can compare the artificial neural response patterns to the brain. Now, the reason why I have a two-way loop is that actually not all ML algorithms will match the brain equally well. In particular, what we'll often need to do is to design ML algorithms to perform the organism's behavior under the right constraints, by identifying the right constraints. And when doing so, this iterative process of better matching the brain through optimizing on a task yields two types of outcomes. One is a quantitatively accurate and practically useful brain model. In other words, if you train an artificial neural network and find that it recapitulates the brain well, you can then go and deploy it in new settings and much more quickly than actually running the experiment in the brain. And from a scientific point of view, it also gives us principles of why neural responses are as they are. In other words, if we optimize a neural network for a task and the response patterns end up quantitatively matching the brain, then that's a strong normative hypothesis for the evolutionary and developmental pressures that brain area that gave rise to those neural responses in the first place. Now, we're not claiming that the brain is always optimal or that evolution is always maximizing, but insofar as neural response patterns are due to selective pressures under a task constraint, then a task-optimized neural network model of that will be useful for finding what those pressures are. And what I'm going to do throughout this talk is use this recipe and apply it to three problems in the perception action loop. In particular, I'm going to study the role of recurrent processing during object recognition. And then I'm going to go to a higher cognitive ability in primates, studying mental simulation, or the ability to predict the future state of the environment a few seconds out, getting closer to action. And then I'm going to cross the species barrier and show you that this task-optimized approach can also apply to interrogating natural intelligence in rodents, in both sensory and non-sensory areas. And then finally, I'm going to close this perception action loop and go into future directions of how to do so by building embodied agents to engage with whole brain data that's coming online now. So just to start with the role of recurrent processing, this was work that was actually done over the course of a number of years with many collaborators, and started as a modeling paper in NeurIPS, and then later a comparison to actual brain data from Jim DiCarlo's group, specifically data that was collected by a co-car that came out in 2022. And this will primarily involve actually mostly modulating the knob of the architecture class or circuit in terms of the questions that we're asking, holding the other things fixed from the CNN picture that I showed you earlier. And the motivation for us was that actually recurrent connections are a quite ubiquitous neuroanatomical phenomenon. So a natural question that arises is, what role do they play in behavior, if any? Now, this is a very big question. So to bite off a piece of it, what we did was we tried to understand this in the realm of rapid visual processing. So the first 250 milliseconds of visual processing. Now, I showed you this picture of how convolutional neural networks, or CNNs, are good models of primate object recognition. They're both functional and structural models. But a major downside is that they actually lack the recurrent connections to enable us to engage with this question meaningfully. And in particular, these recurrent connections, this isn't just biophysical, they play a functional role as well. So Ko had found that actually these recurrent connections enable you to enable primates really to recognize hard images, so these red images, which are hard for feed-forward models, but are decoded later in time, actually by IT. So in other words, the red CNN not solved challenge images are actually solved by the primate ventral stream later in time. by way of recurrent connections. So given that we know that it has some functional relevance, we want to better understand really the neurobiological puzzle of this section is what is the role of recurrent processing in the primary ventral stream during object recognition? And so to get at this question, it becomes clear and very prudent to then start to design ML algorithms to perform the organism's behavior under the constraints that we know that the organism faces. Now, this work was done in 2017, back before vision transformers existed. But this problem of lacking recurrent connections would still persist for vision transformers today. So to really engage with this question, what we did is that we enlarged the class of CNNs to convolutional recurrent networks, or conv-RNNs. And what this consists of is implanting in a CNN both a choice of local recurrence, which I'm highlighting in green here, as well as long-range top-down feedback connections. And furthermore, the way we unroll these dynamics is to treat each time step layer by layer as a 10 millisecond pass, just as it would be in the ventral stream when it's processing a visual stimulus. And the one thing that I want to emphasize is actually that because we're enlarging the class of models, it actually turns out that many choices of recurrence fail to be performant on ImageNet. And in fact, it took us about 1.5 to 2 years to really nail down just good choices of recurrence that could at least just perform the object recognition behavior to begin with. So it became really an architecture problem. Specifically, if we started with a shallow feed-forward model like this BaseNet on the left, and tried to basically climb to get to the performance of a doubly deeper ResNet34 that's maybe closer to human performance on ImageNet, what we found was that actually just implanting kind of standard convolutional versions of recurrent circuits that existed, like LSTMs and GRUs, et cetera, would kind of very barely improve the top one ImageNet accuracy, but they would also add lots of parameters too. So this led us to actually invent our own convolutional recurrent circuit known as the reciprocal gated cell, or the RGC. And this itself actually was inspired by the cortical microcircuit in that it incorporated both the bypass as well as the feedback later in time that we see, the reciprocal gating that we see in the cortical microcircuit. And this motif actually enabled us to not only get much better ImageNet performance, but at the cost of way fewer parameters. Now, I've so far just talked about local recurrence, but what about the other component of long-range feedback connections? Well, to get at this question, since there's lots of different combinations of how you can connect, of different patterns of long-range feedback connections, and then how you also incorporate them with feed-forward input, we did a large-scale search over these, and we parameterized those choices of the operations of combination as well as where they interface in the local recurrence cell. And in collaboration with Google Brain, who very generously allowed us to use their TPU resources, which was very necessary for the time because GPUs were very small, actually, in memory. And these recurrent cells, especially in a deep CNN, take up lots of more VRAM. What we did was ran a large-scale evolutionary search both over the discrete choices of architecture and long-range feedback connectivity, as well as the continuous hyperparameters of learning. So each dot that you see here is a conv-RNN that's being selected for in a Bayesian hyperparameter optimization, and is being trained for 5 epochs on ImageNet. And what you see is after several thousand models, we start to see a plateau in performance and basically an increase over where we initially started. And in fact, the one nice thing about doing these large-scale evolutionary searches is you can actually look at patterns, not just in just the top model, but across, say, the top 50 models or top 100 models. And what we saw, interestingly enough, was that a sort of pattern of global feedback connectivity emerged, as well as choices of certain operations like depth-separable convolutions, which end up being more parameter efficient in the long run. So going back to this question, what we found was that actually we can approach the performance of these deeper models once you have both this local recurrent circuit, this RGC cell, as well as the suitable choice of long-range feedback connection. Now, we spent a lot of time trying to better get at these constraints. Now, what does this yield? Well, going back to now comparing to brain data, we look at the ability to basically decode images like the primate object recognition does in the ventral visual stream. So here is, for every image, we're going to look at the primate accuracy to decode this image correctly relative to any distractors. So that's what the D prime is indicating here. And we're going to look at the time course in IT when it first solves that image. In other words, the first time that decoding off of IT activity from the animal actually matches that primate accuracy line. And furthermore, because our models have dynamics, we can then look at the first time that matches the primate accuracy. And similarly, when IT does this, is the primate object solution time. So basically, for every number, we're going to have, for every image, we're going to have two numbers: the model object solution time, when the model dynamics solve the image and matched primate accuracy, and the primate object solution time when IT did this and matched the primate accuracy. So looking across about 1,300 images, what we see is that on the y-axis is the object solution time consistency, and on the x-axis is the number of images that are both mutually solved by models and inferior temporal cortex. So our goal is to be on the top right of this plot. And looking across a range of both shallow and intermediate and deep feed-forward and conv-RNNs, what we see is that actually the best match to both, matching the same solved images that IT does, as well as the consistency, the time in which it solves it, are these intermediate depth conv-RNNs. So we wanted to better understand, well, why were these intermediate depth conv-RNNs compared to, say, very deep feed-forward models or alternatives better matching these object solution times in the primate? So what we did was that we reasoned that basically these conv-RNNs that are intermediate depth are satisfying a performance and a size trade-off, where we're measuring size in two different ways. One is a number of parameters, and the other is a number of units. So we can physically think of measuring size as a number of parameters as maybe the number of synapses, and the number of units as a physical measurement of size as the number of neuron bodies. And what we see is if I take the same plot and just color its ability to match the primate object solution time consistency, so the redder you are, the better you are, what we see is that when size is measured as a number of parameters and a number of synapses, is that the conv-RNNs, which are the x's here, that best match the object solution times, aren't really differentiated by size in any meaningful way. In fact, lots of different choices of number of parameters can give rise to similar object solution times. On the other hand, when we measure size as a number of units instead, we see that actually these conv-RNNs of intermediate depth that are best matching the object solution times are actually sort of hugging a Pareto-optimal trade-off between keeping the size down but doing as well as you can with that size on ImageNet. And furthermore, the very deep feed-forward models, which are indicated by the squares here, don't match the object solution times as well, and in fact add incremental gains in top one accuracy, but adding millions of more parameters. So the takeaway, going back to our neurobiological puzzle of the role of recurrent processing in the primate ventral stream during core object recognition, is that by replacing and enlarging the class of CNNs with conv-RNNs, we find that this enables the primate ventral stream to attain high object recognition ability under a physical size constraint, done through temporal rather than spatial complexity, specifically by conserving on the number of neurons rather than the number of synapses. So now what I'm going to do is I'm going to go closer to the action part of this perception action loop and build on this foundation of rapid visual processing to study a higher cognitive behavior over a slightly longer time scale of predicting the future state of the environment over the course of a few seconds. And we're going to still stay in the same species of primates and humans. So this is work that actually appeared last year as a NeurIPS spotlight. And in particular was done in collaboration with Mehrdad Jazari's group, where the neural data from primates that we're comparing to here was collected by Rishi Rajalingam. And since a lot less is known about this type of behavior and the brain areas that are involved in it from a computational point of view, it actually will end up modulating the three of the four knobs of the task, architecture, and the environment, unlike the first section where we just modulated the architecture. And we're going to use the learning rule as a proxy, an efficient proxy to get to the adult state of the animal. So the motivation is as follows, is that, you and I can not only rapidly recognize the objects in the scene, but we can also make inferences over longer timescales that go beyond the pixels that are rendered here. So for example, we can infer that the ice block on the right has been out longer than the one on the left, just based on our knowledge of temperature and physical dynamics. My cat will predict whether this box will support her or not before she gets on. And I can also plan intervening actions just by looking at these hats of how I would take them off the rack. And this isn't something that's restricted to static scenes, but also dynamic ones as well. So for example, if I show you this fluid that's coming into contact with this unknown substance, you can actually infer that it was a staircase that was there, even though you weren't shown it visually. And the crux hypothesis comes back to, of how we do this, comes back to the very prescient work of Ken Craik, who's a Scottish psychologist. And this is actually an excerpt from his book, which I really like, which is that the brain builds mental models of the external physical world that supports physical inferences via mental simulations. In other words, the organism carries a small-scale model of external reality in its head. And I want to point out that he actually did think of this as a computation. The symbolism is largely of the same kind as that which is familiar to us in mechanical devices that aid in thought and calculation. This is said, before the modern digital computer. There was only analog devices around at the time. Now, only in the past decade, we started to get evidence for this hypothesis. First from human behavior, and in particular in the domain of physical simulation. So when objects come into contact with one another. And this in particular is the situation we focus on too, because it's where you can get very nice ground truth rather than say emotional affects or other things that can be involved in more complex mental simulation. So most notably the work from Josh Tannenbaum's group with the Intuitive Physics Engine, or the IPE, whereby they show that this sort of probabilistic inference engine that resembles the simulator you see in modern video games can match very well human judgments of whether or not this block tower can fall and in what direction. In particular, unlike video game simulators, models things with uncertainty as well. Now going finally into the brain, so from behavior now into the brain, at least at the level of human neuroimaging, the work of Nancy Kanwisher's group has shown that the frontoparietal network, or the FPN, is likely implicated in this mental simulation ability. So they seem to be recruited by physical inferences, so more by whether or not the block tower will fall or in what direction, rather than just the color of the blocks alone. That kind of visual information contains information about mass, as well as physical stability that can be read out. Now, this is at the level of neuroimaging, which deals with blood oxygen activity, but only in the past year or so have we started to get recordings of spiking activity in the brain, particularly in non-human primates, from the work of the group of Mehrdad Jazari. In particular, in one node of the FPN, known as DMFC, or dorsomedial frontal cortex. And they showed that by training a monkey to play a game of Pong, where the catch is that the ball goes behind the occluder right before it intersects with the paddle, they see evidence that DMFC is actually simulating the ball's position and velocity while the ball is hidden from view. So now I'm interested not just in Pong, but in all of these various settings that we're seeing this mental simulation ability for is what are the functional constraints that enable us to predict the future state of our environment across diverse settings? So by functional constraints, I'm going to encode them using this task optimized approach that I introduced earlier. And we're going to first start by designing ML algorithms to perform the organism's behavior under the organism's constraints. And this is going to be done by modulating these three knobs. And it's going to give rise to a few sensory cognitive hypotheses classes. So models that operate from realistic, naturalistic video inputs to predicting the future state of their environment. So the first broad class of models is going to be what I call end-to-end future prediction. Next would be based on object slots. And finally, the last one is going to be known as latent future prediction. And I'm going to explain that in a minute. Now, by diverse settings, the way we're going to get at this is we're going to actually, because these models are stimulus computable, we can actually evaluate them both on new scenarios for human behavior, as well as the completely out of distribution scenario of Pong, where we can also look at neural recordings as well. So, but we're going to be constrained by both. And there's kind of trade-offs with each, right? Where in the human behavior, it's 3D scenes, but not as detailed neural activity. Whereas in the Pong, we have very detailed neural activity, but a less rich scene, but nonetheless allows us to separate models. So I'm going to explain each of these hypotheses now in the context of matching the neural data. So in particular, on the y-axis is neural predictivity, or the ability to predict dorsal medial frontal cortex activity. And the first thing we did was actually look at how well do two animals, since we have recordings from true primates, DMFCs match each other. And this is known as the animal-to-animal or inter-animal consistency. And we see this is quite high. Now, another thing we could look at is the perfect simulation oracle. So this is basically if you had ground truth access, which the animal doesn't have, to the ball's position and velocity, how well could that predict the neural activity in DMFC while the ball is hidden from view? And we see actually a substantial portion of the neural activity in DMFC is due to this, to simulating the ball, basically. And this allows us to isolate that versus, say, other things like attentional or saccade demands that the animal also has to do that maybe aren't relevant to simulation alone, and that our models, aren't modeling explicitly. So the first class is this end-to-end future prediction, and basically what it consists of is taking in the video at one time step, and then predicting what the next time frame, what the frame will be. And so it has a pixel-wise objective, so it's trying to match the pixels of its output, of its predicted output, with the actual next frame, as well as a KL divergence term to prevent overfitting. And so the thing I want to emphasize, though, is that this predicts the future at the individual pixel resolution of the next frame. So it's very detailed. And the interesting thing was that as we took a variety of these end-to-end pixel-wise feature predictors, trained on a variety of image augmentations as well as image video sizes, we see that all of them poorly predicted DMFC. In particular, if we took the kind of like state-of-the-art ones like Fitbit and tried to assess their generalization, one thing we found is that actually if we gave it the Pong videos and then had it simulate what's going to happen next, it's actually turned out that it treated the ball as though it was static. It didn't recognize that this was a moving object. This held true even if you did a bunch of tricks to help this model, just to show that we're not strawmanning it, is that, it can actually do well on out of distribution, on new scenarios that are in distribution in the videos it's trained on. But in this out of distribution scenario, even if you enlarge the ball to be much bigger, just to make sure it's not something simple like that, it still fails to do this. So this is indicative that it's actually strongly overfitting with its pixel-wise loss to the statistics, as you might expect, of its training environment. And it's not able to do this kind of structural out of distribution generalization. And so it's sort of akin to if I was trained on lots of eons of video data, and then I was put in a new room with new objects that I suddenly fail to recognize which ones are moving or not. That would be very detrimental to the survival of me and any other animal that has this. So this led us to think that perhaps fine, it doesn't predict at the level, this very detailed level, because it fails to generalize. But perhaps then DMFC is predicting a factorized version of the scene. So how might it go and do that? Well, one idea would be to kind of factorize the scene at the level of object slots and their relations. And so a very prominent kind of self-supervised model of this is known as the CSWIM model. And the basic idea is to encode the scene as a level of fixed object slots with a self-super and then train future predict using a graph neural network. And the self-supervised objective is to predict at the next slot what the representation of the scene will be, and then repel that from the kth object slot representation in another video. And so this is done completely in a self-supervised way without labels. But again, what we saw is that even with varying encoder sizes, none of these object slot models predicted DMFC all that well. So if it is predicting a factorized version of the scene, it doesn't seem to be doing this by allocating fixed object slots. So this brought us to the next hypothesis, which is known as latent future prediction. And so rather than posit some explicit, kind of bespoke representation, what we thought was to instead learn a partial implicit representation of the physical world by first performing a challenging vision task or a foundation model, and then future predict in that latent space. So rather than predicting at the level of object slots or pixels of the frame, to do this to hold fixed the vision foundation model and future predict in the latent space. So then the crux question becomes, well, what vision task should we then pre-train on to learn this suitable latent representation? So the first hypothesis class to try is kind of static image categorization that I showed you in the first section. So just simply categorizing on ImageNet what these objects are. And what we found was that actually across a range of image foundation models, even those that are larger than ImageNet, like 400 times larger, none of these matched DMFC well either. And so notably, they're useful for downstream image segmentation, classification, static tasks. But the thing to keep in mind is that we do far more as humans and animals than engage with static scenes, right? In particular, we tend to engage with scenes like these. So this is a data set known as Ego4D that came out in the first year of my postdoc when I just started. And it consists of participants around the world wearing head-mounted cameras and going about their day doing everyday actions. And you can actually train in contrastive self-supervised way on videos to learn representations that attract at time points of video frames representations that are closer together in time and repel from those video frame representations that are earlier in time, as well as on the same batch but from different videos. And it was shown from the work of several people in 2023 that doing this kind of contrastive learning on Ego4D gave rise to representations that could transfer well to out of distribution and body tasks, not perfectly, but much better than, say, what we had with static image foundation models. And so if we take these video foundation models now and compare them, which are useful for a body task, we see that they best match DMFC, approaching this kind of perfect simulation oracle. Now, it's not just enough to pre-train a latent. We also need to do this explicit feature prediction. In other words, if we compare just to the latent alone, the latent isn't enough to do it. In other words, just pre-training on Ego4D isn't a known. You do actually need to explicitly feature predict in that latent representation. So going back to this, now that we've kind of isolated these constraints, what does it give us? Well, like I said, we want to look at this across diverse settings. So we also wanted to look at held out behavior on more naturalistic scenes, where we can collect human behavior. So this is the 3D world, which focuses on everyday physical understanding. that was collected by folks like Dan Baer and Dan Yeamans, Judy Fan, and collaboration with Josh Chennabaum's group. And it consists of 8 different scenarios that involve both contact between rigid objects, but also soft objects like this soft and rigid objects like this grape scenario on the right. And the main readout that humans are queried with is whether the red agent object will come into contact with the yellow patient object. So an example scenario like this, where humans look at this, is the red domino, the agent object, and then here's a side view, clicking yes or no, whether it'll come into contact with the mat before the scene unfolds. So it's a sort of high order readout of what humans will think the simulation will result in. Now, given that basically, macaques can operate in 3D environments, right, very naturally, and that humans can play Pong, there's clearly a type of like species conserved everyday physical understanding that can occur across both humans and non-human primates. And so to better triangulate the models and the constraints, what we did is we evaluated models simultaneously against both measures, because they're stimulus computable, of Pong, of detailed neural activity, and the human readout of contact on held out rich naturalistic scenes. So on the y-axis, again, the ability to predict DMFC in the artificial neural network responses. On the x-axis, the ability to match human behavior in fission. And what we see is that actually, if we treat each metric equally, where the dotted line measures that, what we see is that while no model can perfectly match them both, we see that dynamically equipped video foundation models can do this. And so, in other words, specifically the one that has been exposed to the largest variety of egocentric video sources and transfers the best across the widest range of embodied tasks. And in future directions, I'm going to talk about how we can improve these to get closer to the upper right. But coming back to the neurobiological puzzle for now, what are the functional constraints that enable us to predict the future state of our environment across the first settings? By varying these 3 knobs, what we found is that, and identifying what these constraints are that best match it, is that the brain's mental simulations crucially involve explicit future prediction of some visual scene description. And specifically, this visual scene description used by the brain doesn't appear to be fine-grained at the level of pixels, but it must be factorized somehow. And this factorization in and of itself appears to be strongly constrained. So it doesn't appear to represent fixed object slots, but rather a critical component is for it to enable a wide range of embodied abilities. So finally, what I'm going to do is I'm going to cross the species barrier and show you that this task-optimized approach also allows us to study natural intelligence in other animals as well. So this part of the section will involve modulating all 3 of these knobs, using and holding fixed the learning rule again as an efficient proxy to get us to the natural state of the animal, to the adult state of the animal, and comes from one work that appeared last year in PLOS Comp Bio and collaboration co-led with my collaborator Nathan Kong, as well as work that appeared a few years ago not in rodent visual cortex, but now in rodent medial entorhinal cortex, so a non-sensory area. That was NeurIP Spotlight that was done in collaboration with Lisa Giacomo's group with experimental data compared to there. So going back to the first picture I showed you in this talk, I showed you that CNNs are functional and structural models of primate object recognition. And I'd shown you this kind of nice alignment between a neuroscience goal and an AI goal when it came to the primate ventral stream. But what about other animals, like, for example, the visual system of the mouse? Well, already purely from looking at the neuroanatomy, a mouse brain area, like especially its visual areas, are way less hierarchical. So in other words, it kind of looks like a paw, where V1 is sort of the largest area, and then you have a bunch of higher order visual areas that are kind of rest on top of it in parallel. In fact, if we make this exact same plot of neural predictivity versus top 1 validation accuracy, what we see is that actually we don't see a strong correspondence between doing this task and predicting neurons. And in fact, the better of a image categorizer you are, the worse of a mouse model you become. So the natural question is does task optimization apply to rodents? And what I'm going to show you is that, in fact, the answer is yes. And it really does come down to the fact that the constraints that we have with these primate models are very different than the constraints that the actual mouse visual system has to deal with. And so it'll be prudent to identify what those constraints are. And long story short, what we found was that shallow contrastive CNNs are actually best matching the mouse visual system. So in other words, unsupervised learners that were very powerful and supervised algorithms that came out in the last several years were very crucial, coupled with a shallow visual cortex that could do this. So we wanted to understand, why is this the case? Why is, in particular, why is this approaching actually the animal-to-animal consistency is about 90% of this data noise ceiling, actually. So why might the mouse visual system be preferring self-supervision? And the hypothesis was that actually it's enabling to learn a task general representation under the resource constraints it's dealing with, of a shallower cortex operating at lower visual acuity, rather than specializing on any one task. So to assess this, what we did was we train our mouse visual cortex on our pre-training data set, a lower resolution version of ImageNet, and then we evaluate on a completely new out-of-distribution task like reward-based navigation. Now, not only is this out of distribution in the sense of like the images are very different, but the task itself is very different in kind too. Specifically, what we do is we embody this mouse visual cortex. In particular, we pre-train it on ImageNet, and then we implant it, we fix the weights, and we implant it in a biomechanically realistic rodent body, which itself is high degree of freedom. And then we train a policy on that vision network to control this body to get this reward, the blue orbs that you're seeing. And so what you're seeing actually is, after training with the resultant policy, what it looks like from the egocentric point of view of this virtual rodent. And this is a challenging, actually, RL problem because it's biomechanically realistic. There's lots of degrees of freedom, and furthermore, you have to do this navigation task. You have to keep track of history over long time scales with high-dimensional continuous inputs. And what we see is that actually, if we take various choices of this visual cortex and train an RL policy on top of it to do this embodied virtual rodent navigation, we see that the contrastive models, the ones that best match mouse visual cortex, better do on this reward-based navigation task than the category specialized models in black here, where the y-axis is the mean episode return. Now we can go the other way. So pre-train in a maze environment and then evaluate on a variety of visual scene understanding tasks at the visual acuity of the animal. And again, we see that the contrastive objective better matches the supervised counterpart, where the supervision signal in this case in the maze environment is the reward. So across a range of these visual scene and understanding evaluations. So that's a trend that we see overall, is that the contrastive models yield better transfer performance across a wide range of statistics, but also tasks as well. Now, going back to navigation, here is a top-down view of a rodent in an open environment where the red dots are place cells in its hippocampus. And these place cells are firing as it enters a particular location, forming a place field. Now, the hippocampus and place cells in particular aren't the only brain areas and cell types that are actually implicated in this ability for rodents to navigate. But also a brain region known as medial entorhinal cortex plays an important role. And this consists of a variety of cell types. So the most prominent and famous ones are these grid cells, which fire periodic fields across space. Now, this is actually in data that I quantified that was collected from by Caitlin Mallory and Lisa Giacomo's group. But this 22% is actually a very liberal estimate. In fact, it's really more like 2 to 3% if you really want the cells that look very pretty, that are on the cover of Science or Nature. Now, another, say, 10 to 12% consists of these cells known as border cells, which have very interpretable response patterns in which they fire at one of the four corners of the box that the animal's in. But a large swath of these cells are actually known as, you could call them heterogeneous cells, and that they don't have a very interpretable response pattern as a function of space. And furthermore, they also encode for a wide range of different navigational variables, from head direction and running speed and just position alone. So this brings us to the neurobiological puzzle is, well, if a large fraction of these cells just exhibit very non-spatially or non-navigationally interpretable response patterns, how might we characterize what these heterogeneous cells do? And in particular, are they noise, or are they actually like functionally relevant? And what role would that be, if any? And to kind of make a long story short, what we found is that actually these heterogeneous cell types emerge in unison with the grid cells and the border cells in networks that are optimized for path integration. But if we take path integration literally and just try to basically integrate position over time, integrate velocity over time to get to the next position in Cartesian space, what we see is that actually these networks are actually too biased towards the heterogeneous cells, and they don't actually develop these grid cells. But furthermore, the animal it doesn't even have access to ground truth XY position anyway, but it does have access to the place cells in the hippocampus. So implanting these gives rise to the heterogeneity in this exact form, without the exact proportions that we're showing here, without explicitly going for it, but just purely optimizing for this place cell based path integration objective. So now that we've isolated the constraints, what are the outcomes? Well, our puzzle was, does task optimization apply to rodents? And by isolating these constraints, these three knobs, to better figure out what they are, what we found, at least in mouse visual cortex, is a shallow, low resolution, contrastive, self-supervised objective that gives us the functional understanding of mouse visual cortex as making best use of the mouse's limited resources to create a general purpose visual system. And furthermore, going from a sensory area to a non-sensory area in the domain of navigation, by using this task optimized approach, we find that grid cells aren't uniquely relevant to navigation. And in fact, both heterogeneous cells and grid cells arise jointly through task optimization. So just to recap before I go to future directions, what we covered in these three sections, when it came to the role of recurrent processing during core object recognition, we find that it enables the primate ventral stream to attain high object recognition ability through temporal rather than spatial complexity, specifically conserving a number of neurons as our measure of space. We then studied higher cognitive behavior over the next few seconds of predicting the future state of the environment. And what we find is that mental simulation crucially relies on explicit feature prediction of a factorized description of visual scenes, where this factorized description is strongly constrained and must enable a wide range of dynamic sensory motor abilities. Then we crossed the species barrier and showed that in both mouse visual cortex and rodent entorhinal cortex are best explained by a biological performance optimization on a suitable task objective. So now what I'm going to do is turn in the time remaining to future directions, where I'm going to extend the task optimized approach to close this perception action loop by building embodied agents to engage with whole brain data that's coming online now. So throughout this talk, I've just kind of shown you this basic recipe of task optimized modeling, where we try to identify what these constraints are, and then it yields meaningful and useful outcomes, as well as scientific insight of why neural responses are as they are. But so far, what constraints are we still missing, when doing this? Well, I modulated these four knobs and showed you that there was applicable to provide insights across a variety of brain areas and species. But so far, these are models of individual brain areas, and none of them together form an integrative understanding of any one organism's brain. And I would argue that we also, don't have a complete understanding of any individual brain area if we don't also study how it interacts with other brain areas as well. And this is especially prudent as with the advent of neuropixel recording technologies are starting to gain access, really detailed access to whole brain recordings from awake behaving animals, most notably rodents in this case. So a pressing need arises of how we're going to make sense of all this data, especially as it affords us a unique opportunity to give us insight into brain function. And so I think it's going to be crucial to be able to reverse engineer the animal brain and really provide a systems level understanding of the brain in precise engineering terms. So how might we go about doing this? Well, I think to do this, we need to start by getting serious about building artificial organisms rather than sticking to kind of the individual brain area models that we've had thus far. And so just to keep the discussion focused for now, I'm going to focus on working towards building artificial rodents where we really are starting to get this whole brain data from to be able to explore these in a grounded way. So in particular, what we want to work towards is an integrated task optimized model of the rodent. You might ask why. Well, one, it's the de facto organism of choice in neuroscience. So that's where we're getting this whole brain data from to be able to, in a detailed way, understand brain function. But from a natural intelligence point of view, rodents also perform very interesting embodied behaviors. They can navigate and plan. They exhibit flexible and adaptive motor control. And interestingly, they can both display autonomous and also trained decision making. So why am I highlighting embodied here? Well, this is actually a domain that current generative AI with autoregressive losses trained on massive data sets actually really fails to do well. So in the past half year or so, a very prominent kind of version of this is Sora, released by OpenAI, that generates very high fidelity videos from text inputs. And here's an example of one where if you look at this video, you see this kind of chair you just come out of nowhere. And not only that, but like the physical dynamics are wrong. Just as you touch it, it just kind of floats in this, really unphysical way, right? And also fails to maintain object permanence too. So a natural question is, well, what's missing to really extend this further? And I would argue that really we need not just passive training on videos, but really to sort of model embodied interaction to get this common sense physical understanding. So how are we going to do this? Well, I'm going to split this into two directions. One is building the embodied agent, and in the second direction, applying the embodied agents to neuroscience questions. So starting with the first direction, we can actually take what we have. They sort of form, disjointed pieces of what we want, and start to fit them together into an agent. And first of all, before doing this, the overarching question that this gets us is, how does the brain build and use world models? And I like to think of world models as four broad components of an animal. How does it represent, predict, plan, and enable action? So we can think of the work that I've shown you so far in primate and mouse vision, in the first part of my talk, as forming the represent part of this sensory motor loop. The second part of my talk is dealing with the predict part. The third part is starting to engage with planning. And then finally, a kind of common theme across all of these is that, if you have a good neural model, you also are able to do better sensory motor control as well. Right? That was a kind of a common theme that arose across these, setting us up. But to really connect these together, I think we need to improve each of these components in a variety of ways. So specifically, I think we need to go beyond the static images that I showed you in the first talk, and study both the role of recurrence and self-supervision to enable sensory learning in dynamic environments. And the reason I focus on this combination of recurrence and self-supervision is that recurrence is also not only do animals not get external labels, so hence the need for self-supervision, but recurrence is also a very ubiquitous neuroanatomical phenomenon. So perhaps this might be a repeated motif and principle that's useful for enabling also multimodal learning, like audition, olfaction, tactile perception, et cetera. And that would underlie both more robust feature inference, such as, for example, extending the work we did in the second part of the talk by more explicitly representing objects, as well as representing multiple timescales to be able to deal with multiple possible future possibilities. And going beyond the kind of simple environments we did when we were studying hippocampus in the third talk, by enabling longer range planning in more complex, visually rich environments. And a kind of natural starting point is just to connect the mouse visual cortex in silico that I showed you with the hippocampal models that we've built. And furthermore, we just engage with MEC, but what about hippocampus? Hippocampus doesn't just consist of place cells, but there's heterogeneity as well. So by having a more end-to-end process with a visual front end, can that already engage with the heterogeneity problem in both MEC and HPC? And finally, it's known just from neuroanatomical observations that motor cortex consists of very highly stereotyped neural circuit motifs of both hierarchy, but also recurrent processing. So can some of the insights that we found in constrained sensory motor or sensory processing also be useful for enabling more sample-efficient motor learning of biomechanically realistic animal bodies, which have very high degree of freedom and are very difficult to learn with current end-to-end RL techniques? And finally, I want to emphasize that each of these modules, which I'm color coding here in the schematic of the rodent brain that best represents it, will be compared, their internal representations, in the same ways that we've done so far, to real neural recordings from brain areas, for example, in the rodent here. And so the color coding is just meant to represent hypotheses for brain areas that would best be predicted by each of these modules. But the other thing I want to emphasize is that the agent as a whole, namely these schematic black arrows that I'm showing you, are a concrete hypothesis for how these brain areas collaborate to give rise to complex behavior. So these individual brain areas are no longer in isolation. So now that we have the embodied agent, then what can we do with it? Well, it yields naturally a few practical outcomes. The first is, given that more and more open source data, open source data is coming online from whole brains, especially rodents, this would naturally form a set of benchmarks that we could release to the community. And crucially, having these in silico embodied agents allows you to predict outcomes for new experiments. So because this is in silico, you can run millions of alterations on these and perturbations on these models to more cheaply run new hypotheses than you could in the biological brain. And we would already validate in the brain data we have whether these embodied agents are good predictors and representatives of pre-existing brain data and pre-registered brain data. Finally, from just a less engineering but more scientific point of view, it allows us a systems level understanding of an organism. So what are some questions that we can engage with that systems level understanding that we couldn't with kind of individual brain models? Well, the first is that it can actually give us a platform to start to study the hardwired intrinsic goals that enable animals to survive in complex environments. And unlike reinforcement learning in games, like in AlphaGo, for example, these objectives aren't as well specified and are quite underspecified. So having a detailed computational understanding is not only a very interesting point of view from brain science, but it's also going to be very crucial for building the next generation of autonomous agents. Furthermore, we can start to understand how these modules interact to enable learning of a task, and crucially to better identify the learning algorithms that are actually in the brain. So I have a whole body of work that tries to engage with the question of, what might be some plausible learning rules in the brain. But crucially, to really get this learning rules program off the ground, we also need an agent to be able to specify what the right inputs and outputs are to begin with, especially in brain areas where we might want to study that have high degrees of plasticity, like, for example, hippocampus. So having an understanding of the visual input or visual representations that go into hippocampus to begin with is going to be crucial for understanding how learning occurs. The inputs and the outputs have to be well specified, and that's what the agent gives us. Lastly, it also allows us to study species-conserved algorithms across modules. What do I mean by that? Well, this sensory motor loop isn't just restricted to the rodent. It's something that's common and shared across all animals that we're getting neural data from fruit flies to zebrafish to rodents, macaques, and infants. And so here what I'm showing you is the brains of different species, so rodents, macaques, and humans, with their homologous brain areas highlighted. And the main point is that you wouldn't have one integrative agent for across all these species, but rather one integrative agent per species. And what this allows you to do is, in the long term, as an extensible platform, is to be able to study, the evolutionary conserved principles of natural intelligence that are involved in this sensory motor loop in a detailed way, because now we have, these computational modules and connections specified. And the main point of why we want to focus on the sensory motor loop is that most notably, no robot exists today that's as autonomous and adaptive as biological animals. And this is something where natural intelligence truly shines. And as we're leveraging the nexus of two advances here of large-scale brain recordings to constrain our hypotheses and advancements in AI to generate those hypotheses, this is something where neuroscience and AI can learn virtuously from one another. The brain science of the future is going to heavily leverage machine learning to build functional and predictive theories of natural intelligence. And that's what this program is best positioned to provide. With that, I'd like to thank my collaborators, without whom none of this work would be possible, as well as my funding sources. And please reach out if you're interested, and thank you for your time.